Hello, I'm Dr. Remi Creusot and I lead a team of researchers at Columbia University Medical Center to study type 1 diabetes and develop a cure. In this video, I will explain one of the approaches that we are following to stop the immune system from attacking insulin-producing cells. Dendritic cells are immune cells that act as both sentinels and educators. They are always on the lookout for pathogens and they initiate important immune responses by triggering T cells. If a microbe or a virus intrudes, it gets broken down by a first line of defense composed of microbe-degrading immune cells. Among the debris generated, there are two types of microbial fragments. The first type includes antigens, which are further degraded by the dendritic cells and presented on the surface of the cell as something that needs to be targeted. The second type includes products that are perceived as danger signals. These danger signals turn on the immunogenic mode of dendritic cells. Immunogenic dendritic cells are able to instruct T cells that can recognize the microbial antigens to attack the microbes that display these antigens. Therefore, immunogenic dendritic cells are important to stimulate T cells into attacking microbes. Because that's just what they do, dendritic cells also pick up antigens that are produced by our own cells or released from our cells when they die. These are called self-antigens. Although they can present these self-antigens, the lack of inflammation or infection means that these dendritic cells are not turned on. They maintain a tolerance-inducing or tolerogenic mode. This means that when they encounter T cells that can recognize these self-antigens, not only they will not be able to activate them, but instead they will program them to die or to never respond. We all produce T cells that recognize random things. Some may be potential microbial invaders and some may be part of self. This is the duty of dendritic cells to eliminate these self-reactive T cells before they can cause any harm. Therefore, tolerogenic educator cells are also important to rid ourselves from self-reactive T cells. However, two conditions when met in the same individual can lead to autoimmune disease. The first condition is a defective reprogramming or elimination of self-reactive T cells. In other words, either the tolerogenic educator cells lack the necessary signals to instruct self-reactive T cells appropriately, or the self-reactive T cells have become refractory to such instructions. As a result, potentially harmful self-reactive T cells are not being purged or inactivated. The second condition may be the release of self-antigens from injured tissue that is accompanied by inflammation. In type 1 diabetes, this inflammation could be due to a viral infection that leads to the death of some insulin-producing beta cells. In this situation, dendritic cells perceive the inflammation and adopt an immunogenic mode. They also end up acquiring and presenting self-antigens, for example derived from beta cells, thinking that they may be microbial antigens. These dendritic cells can now potentially activate T cells that recognize these self-antigens if they exist. Thus, presentation of self-antigens by immunogenic dendritic cells can be potentially very bad. This dangerous situation is not so uncommon, but is usually avoided. Indeed, in most individuals, the tolerogenic educator cells will be able to catch and block activated self-reactive T cells before they can do any harm. However, we've seen that in some individuals, both conditions are met, meaning that the tolerogenic educator cells are not performing well. This can lead to a high frequency of circulating self-reactive T cells, and once inflammation occurs at the wrong place, the tolerogenic cells may not be able to intercept and reprogram activated self-reactive T cells. which can then launch an attack against the tissue that produced the self-antigen in question. In type 1 diabetes, T cells that are reactive to insulin and other components of beta cells are not properly eliminated, and if activated, they are not properly counteracted. In our lab, we are able to turn white blood cells into dendritic cells. We can then upload information into these dendritic cells in the form of DNA or RNA so that they can start producing molecules that can help them communicate better with self-reactive T cells. These dendritic cells are in this way manipulated to become super educator cells. First, dendritic cells are provided with common self-antigens so that they can interact with the relevant T cells without affecting the rest of the immune system. Second, dendritic cells are provided with molecules that are used to efficiently reprogram these T cells, the kind of information that the T cells cannot ignore. Third, dendritic cells are provided stabilizing instructions, meaning that they will be locked at tolerogenic mode and cannot become immunogenic and inadvertently activate T cells. Depending on the signals delivered by the super educator cells, the target self-reactive T cells will be either eradicated or recruited into an elite squad called regulatory T cells to assist with the control of other self-reactive T cells. These regulatory T cells of the elite squad are able to help other dendritic cells that may struggle to instruct self-reactive T cells. Thus, in addition to the super educator cells injected, the patient's own tolerogenic dendritic cells will become better at blocking harmful self-reactive T cells that are mistakenly activated by immunogenic dendritic cells, including inactivating them or reprogramming them to become harmful regulatory T cells. Your support and contribution will provide seed funds that will kickstart a new project in this area towards creating super educator cells. Seed funds help researchers in generating the initial data needed to apply for federal or foundation grants. Your donation through Consano is tax deductible and is directly used to support our research without overhead. Please help us build an army of super educator cells to reprogram the immune cells that cause type 1 diabetes.